Hello, everybody. I am teacher Natasha, and today let's talk about cell biology. Before we talk about cells, let's take a look at what equipment we used to take a look at cells. So we know that cells are very, very tiny and we cannot see them through our naked eyes. So that means we need to use a special equipment called a microscope. So before we go into the parts of a microscope, let's see how do we hold a microscope properly. So in order to hold a microscope properly, first you need to know what is an arm. So this part here, we called it arm. So we take our fingers and then we run it through the arm and hold it like this. With our left hand, we place our left hand over the base to support it. So this way we can hold our microscope properly and it won't fall. See how steady it is in my hands? Right. Now let's talk about the parts. First, we have the eyepiece lens. So this part is called as the eyepiece lens. Then, which is connected. So this is the part which we see the specimen from. So that's connected to three of these lenses. These are called objective lenses, and it has three different powers. And this is the stage. This is the area we keep our slide on. And then we have these little two stage clips which secures the slide along with a power point. So this is the light source. So this is an electrical microscope. We have a light microscope as well, which we use normal sunlight to observe our specimens. And then here we have the base. We also have a coarse focusing knob, and unfortunately for me to show you, this does not have a fine focusing knob, but here this part is known as the fine focusing knob. And then here you can see the switch to on and off the lights from the microscope. And then this part down over here is called a diaphragm. So this part, which allows and limits the light entering from the light source. So this is how a microscope looks and the parts of a microscope. Right, now that we know the parts of a microscope, let's move on into how to calculate magnification. So the equation to calculate magnification is image size over the real image. In order to calculate the image size, what we do is we take a ruler and measure either the width or the height of the image that they have given. And in order to find the magnification, you divide by the real size which they have given as well. So when we are doing that, we come across certain conversions in units. So for example, if you want to convert centimeters to millimeters, you have to multiply by 10. If you want to convert micrometers to millimeters, you have to divide by 1000. So if you take a screenshot of this, easily you can work out between the conversions. Right. So you can pause this video and try your own to do these questions. Let's go through these. First question, write down the equation for calculating magnification. So we know the equation. What is it? Magnification is equal to image size divided by the real size. Next, we have a cell with a real size of 0.05 millimeters, which is viewed under a light microscope. The image size is 40 millimeters. Calculate the magnification. Then what do? We put these values into the equation that we know. So it's 40 divided by 0.05, which is the real size. We get a magnification of 800. Next, convert the following. 100 millimeters to centimeters, 100 centimeters, 8,400 nanometers to micrometers, 8.4 micrometers, 0.009 millimeters to nanometers, 900 nanometers. So if you got all of these correct, give a small pat on your shoulder. You have done it well. All right, now let's talk about eukaryotic cells. So we have two types of cells, eukaryotic cells and prokaryotic cells. So what's the difference between this eukaryotic cell and a prokaryotic cell? Well, let me tell you. Eukaryotic cells, they are enclosed. Their genetic material is enclosed inside a nucleus, whereas prokaryotes, that's not the case. They do not have a true nucleus and they have plasmids, loops of DNA inside them. So talking about eukaryotic cells, two of the famous example that we talk about is a typical animal cell and a typical plant cell. So there are components that includes both for animal and plant cell, and there are some components that is only included in a plant cell. So let's talk about what are these components. Right. So the components that are similar or that are same and available in both cells are nucleus, which we know all our DNA, our genetic material, our hair color, eye color, all of those genetic materials are enclosed in nucleus. And then we have cytoplasm, a jelly-like structure where it allows chemical reactions to take place. Then we have ribosomes where protein synthesis occurs. And then cell membrane, this outer covering of the animal cell and this inner covering of the plant cell. So cell membranes allow certain substances to go in and out of the cell. Then finally, we have mitochondria. So mitochondria is also known as the powerhouse of a cell. That is the point where respiration occurs. And there are certain components, like I told, which are specific for the plant cells. They are chloroplast, cell wall, and vacuole. So vacuole, it contains the cell sap. Cell wall, it gives the rigid rice shape for the plant cell. And chloroplast is where photosynthesis occurs. Right, let's talk about prokaryotic cells. So the cell here that we have taken is a bacterial cell. Similar to plant cell, the bacterial cell also have ribosomes, cell wall, cytoplasm. And like an animal cell, a flagella. So do you know what animal cell has a flagella? Yes, it's the sperm cell. So unlike both of the cells, in a bacterial cells, you have plasmids and DNA loops. All right, so again, you can pause the video and answer this question. Well, let's see. So a nucleus contains genetic material and controls the cell. Cytoplasm, it's where chemical reactions take place. Cell membrane controls the movements of substances in and out of the cell. Mitochondria, site of aerobic respiration. Ribosomes, site of protein synthesis. You know proteins are very essential to us. Cell wall supports and strengthens the cell. Vacuole, store of cell sap. And finally, chloroplast, site of photosynthesis. Next, another easy question. So pause the video and do it yourself. All right, so let's see. Nucleus is present in animal cells and plant cells only. Cytoplasm is present in all of the three cells. Chloroplast, only plant cells. Cell membrane across all three cells. Permanent vacuole, only in a plant cell. Mitochondria in both animal and plant cells. Ribosomes across all three cells. and finally cell wall in plant cells and bacterial cells. So you can answer these questions and comment down below. Okay, thank you, my dear students. Until next time with another video like this.